Hello, everyone, and welcome to another edition of Contagion. My name is John Parkinson, and I'm the senior editor. Joining us today is Dr. Tim Rodell, Chief Executive Officer of Aethlon Medical. We are discussing his company's device that is being looked at for COVID-19 treatment. Thank you, Doctor, for being able to join us today.
Thanks, it's great to be here.
Absolutely. And we'll jump right into the questions. Can you provide a brief overview of your company and what it does?
Yeah, sure. So Aethlon is a small, publicly traded company based in San Diego, and we are primarily focused on developing a proprietary, unique device for a number of different indications. The device is called the Hemopurifier, and it's an extracorporeal cartridge, meaning that it filters blood outside the body, similar to a dialysis cartridge, but it has some unique properties. It starts out as a fairly stock plasmapheresis cartridge, which is a cartridge that can be used to filter the blood and take off plasma. For instance, the plasma donations that are being used now in COVID patients from previously infected patients are actually harvested using a plasmapheresis cartridge. But what is unique about the Hemopurifier is that plasmapheresis cartridges are filled with hollow fibers, and the blood flows through the fibers, and the fibers have pores in them that can be of various sizes. And the pores in the fibers in the Hemopurifier are 200 nanometers, or 200 billionths of a meter in diameter. And what we've done is we've surrounded those fibers with a substance called a lectin. And lectins are proteins that are found, they're ubiquitous in nature. They're found in plants, they're found in animals, they're found in bacteria. They're pretty much everywhere. But the lectin that we're using is called GNA, and it comes from the root system of the snowdrop flower, which you may have growing in your backyard, depending on where you live. But the characteristic of GNA is that it binds very tightly, essentially irreversibly, to sugars, and a specific sugar called mannose, which is found on the surface of a number of different biologic membranes. So what that means is that as the blood is filtered through the cartridge and through these fibers, along with the plasma that can flow out, anything that is under 200 nanometers can also flow out. And if that particle has mannose on its surface, it will bind irreversibly to the GNA lectin and be cleared from the circulation. The two particles, relevant biologic particles, that fit into that classification are virtually all viruses. That is, virtually all viruses that affect humans are glycosylated, meaning they have sugars on their surface, and the sugars they have are mannose. So any glycosylated virus, and as I said, that includes all of the major virus families that affect humans. So herpes viruses, of which there are a number, influenza virus, and importantly, coronaviruses, all have mannose on their surface and are all under 200 nanometers. Now we're talking about viral disease today, but there's a critical additional particle that the Hemopurifier is capable of clearing. And that is a small subcellular particle called an exosome. And exosomes have been known for many years. It's been recognized that they circulate in a number of different situations. But for many years, up until about 12 or 15 years ago, they were thought to be basically cellular trash, sort of like cellular dandruff, if you will. But what's now increasingly being understood is that exosomes are one of the primary mechanisms by which cells that are not in direct contact with each other talk to each other. They are the primary means of intracellular communication between cells that are not in contact with each other. And they're shed from normal cells, but they're also shed from malignant cells in cancer in much, much greater quantities. And so because the Hemopurifier can clear both viruses and exosomes using exactly the same mechanism, it has very broad applications in viral disease, but it also has applications in cancer, and we're investigating that, and in transplant, And most importantly, and this is an emerging area right now, exosomes now appear to be critically involved in viral disease, including COVID-19. And I'll talk about that a bit more in a minute.
Yeah, absolutely. It sounds very exciting and very fascinating in terms of this whole way of treating patients potentially. And in speaking of treating patients, there was an example of a Ugandan pediatrician who had been treating Ebola patients and was infected and eventually became comatose and was going into multiple organ failure. There was a remarkable response to a single administration of your device, which helped to save the physician's life. Can you talk a little bit about that case study?
Yeah, it's great, and I'm glad you found that and brought it up. Yeah, this was back in I think 2016, at the height of the last Ebola outbreak, not the one that's currently going on now and tapering off. But yeah, this was a physician from Médecins Sans Frontières who was flown out of West Africa to a hospital in Germany. And the German physicians who were treating him, as you said, he was in extremis. He was on a ventilator, he was on multiple blood pressure drugs, he was in renal failure, and he was comatose. And they had heard about what we're doing and what we're able to do. And they contacted us. This was before I got to the company, actually. They contacted us, and we were able to fly a Hemopurifier cartridge over. They treated him for 6 hours on the Hemopurifier, reduced the amount of circulating virus in his blood by 1,000-fold. And he subsequently recovered. He regained his organ function, and he recovered completely. Importantly, we were actually able to demonstrate that directly that the viral load reduction, the reduction in his circulating virus, was in fact due to the Hemopurifier, not something else, because we could actually take the cartridge and get the virus out of it. We could show that we had cleared the virus from it. So that's, a single patient and a single story, but we had before that treated not a large number, but over 30 patients with hepatitis C, back when hepatitis C was not curable with a single once-a-day drug. And what we were able to show in those patients with about 150 treatments with the Hemopurifier was, number one, that it was safe. We never had a serious adverse event associated with treatment with the Hemopurifier, but also that we were able to clear very substantial amounts of circulating virus. Now, both Ebola and other related hemorrhagic fever viruses like Marburg agent, the hantavirus and others are glycosylated. So is hepatitis C virus. So are all influenza viruses. And in the lab, we've shown with a laboratory version of the Hemopurifier that we can clear multiple different herpes viruses, other hemorrhagic fever viruses. We've looked at 5 different strains of influenza. As you probably know, everybody knew a pandemic was coming. It was just a matter of time. What everybody was expecting was pandemic influenza, like the 1918 flu epidemic. And we've shown actually that virus has been recreated in the lab, and we've shown that the Hemopurifier can clear that. But importantly, relevant to what's happening today, we had also obtained, as you probably know, there have been 2 other life-threatening coronaviruses outbreaks. The original SARS outbreak in 2002, and then subsequently the so-called Middle Eastern Respiratory Syndrome outbreak a few years after that. And we were able to obtain a lab version of the MERS virus, which is a very closely related virus to the current SARS-CoV-2 COVID virus, and we showed that we could clear that very efficiently. So when this outbreak started, we looked at all of that. We looked at our data. We were not able at the time to get the actual SARS-CoV-2 COVID virus in our lab, because that has to be handled under very strict quarantine conditions, which we don't have. But we were able to obtain what's called the spike protein, which is the protein that binds to cells on the virus that allows it to enter cells. And we showed that we could very efficiently clear that protein from biologic fluids. So based on that, the other thing I should say is that after we treated that Ebola patient, we went to FDA and we opened what was then called a compassionate use protocol. It's now called expanded access, so that we could treat any Ebola patients that made it back to the United States or to Canada. We have a similar one in Canada. Fortunately, during this recent outbreak, no patients have made it back to North America. But based on that, we went to FDA and we said, it makes sense. We think it makes sense for us to do a similar thing with SARS-CoV-2, the COVID virus. And so we filed a supplement to our open, what's called investigational device exemption, or IDE. It's the equivalent of what for a drug would be called an IND. We opened a supplement to that to treat COVID-19 patients. And that was done, we got that approved a month or so ago. And we're now in the process of opening clinical trial sites to treat these patients. We're authorized to open up to 20 sites and enroll up to 40 patients. We don't expect to open all 20 sites simultaneously because, as you know, the virus is moving around. So it was first hit New York and New Jersey in the metropolitan area. Now those places have quieted down, partly because a fair number of patients are immune there, but also because people are being more careful. But it's now very heavily in Florida, Southern California, and as you may have heard, as a result of this big motorcycle gathering in South Dakota a few weeks ago, where a lot of people were partying without masks very close together, it's now breaking out in the Midwest. So we're in the process of identifying trial sites and getting them open so that we can begin to treat these patients under that protocol. In the meantime, there is an alternate pathway by which, in an emergency situation, you can treat patients with an experimental device or drug. And that's called the single patient emergency use pathway. And under that pathway, a physician can call us and say, I have a patient here. I've done everything I can. I've given them all of the available treatments that people are using. In this case, plasma, the drug remdesivir, steroids, all of those things, and the patient isn't getting better. And I want to, as a last resort, I want to try your device in this case. We have treated our first patient under that provision, a patient who was extremely sick, who had been treated with everything else. We have not yet disclosed outcomes or anything like that. What I can say, however, is that the treatments went very well. We treated the patient multiple times. And while we're in the process of getting formal trial sites open, we can continue to do that when requested.
Okay, and you had mentioned the study, which was going to lead to my next question. Can you provide, Doctor, some of the specifics of the study, including what you see as the treatment protocol?
Yeah, so we haven't put out a huge amount of detail, but we will be in the next few days. But we will be treating the sickest patients. These will all be patients who are in an ICU. Almost all of them will probably be on ventilators. Most of them, if not all of them, will probably have some renal injury, some kidney injury. So this is going to be the sickest patients. Now, in a situation like this, and it's true in cancer, it's true in a lot of diseases, you tend to start with the patients who really have the fewest options. What I will say is that ultimately, we would hope and expect to be treating patients a bit earlier. And there are a couple of reasons for that. Number one, somebody who isn't as sick has a better chance to recover if you clear the virus from them. Number two, what is not that well understood at this point is what the relationship is between virus circulating in your blood and the manifestations of the disease. What we know from studies in China and other places is that the sickest patients do have virus in their blood, but generally the virus is being diagnosed, it's being detected in nasal swabs in the upper airway, which is the portal of entry, or at least is usually the portal of entry. But there appears to be a very strong relationship between having virus in your blood and poor outcomes. And that's been shown in studies from China and elsewhere. But the other critical point here, and this is where the story about exosomes comes back, is that what we now know is that virally infected cells shed exosomes. And those exosomes have biologic functions. It's been known for some time that circulating exosomes are involved in inflammation. And as you probably know, and as your viewers know, patients generally don't die of the direct virus. They die of the systemic response to the virus. This is sometimes called cytokine storm. And what's now known is that exosomes carrying information, okay, and exosomes can basically carry any molecule that is in the cell. So those could be viral molecules, and they can be nucleic acids, RNA, DNA, and proteins. They are heavily involved in inflammation. But perhaps most importantly for this disease, there's a report out of China that shows that the coagulopathy, that is the blood clotting disorder that is killing so many people, in fact, may be mediated by exosomes. So we not only are going to be clearing virus when it's circulating, we potentially are also going to be clearing mediators of the responses that are killing patients. As you probably know, some patients are getting off of ventilators, being discharged from the ICU, and even going home, and then dying of strokes, blood clots elsewhere, all of these kinds of things. So there's a dual mechanism of action. And in fact, there are a number of other devices that look superficially like the Hemopurifier, that is their extracorporeal cartridges, that are being used in these patients experimentally. But the difference between those and the Hemopurifier is that those cartridges can clear molecules, but they don't clear particles the way that the Hemopurifier does. So we've got a dual mechanism of action, either or both components of which may be critical in these patients.
Absolutely. And I know you said earlier that you're just starting to look for, look into this new study. Do you have any kind of timeline or expectations of when you'll have results for it?
Well, first of all, as I said, we're just opening the sites now. We're just identifying and opening the sites now. So, we hope to have, a number of sites open by year end and be enrolling patients. As I said, you have to go, this is the old metaphor, skate to where the puck is going to be, not to where it is. You have to sort of identify where things are happening. I won't identify the hospital, but I spoke to a hospital in the Southwest last week that they had been getting absolutely hammered with these patients. And for the previous month, and when I spoke to them, they didn't have a single patient in the ICU. So as I said, it moves around. We have to be in the places where we know that it's going to happen. In the meantime, as I said, we can treat individual patients on the request of the physician. Now, one of the things that is, scientifically very interesting about this and will be very helpful is, first of all, this is an unblinded study. We're not treating people, with an inactive cartridge. So everybody is going to be treated in this study. And we can look directly at what we're doing. So we can draw blood before and after the Hemopurifier treatment. We can evaluate how much virus is in it pre and post. We can also evaluate what exosomes are in it pre and post. And then, as I said, we did with the patient with Ebola in Germany, we can actually elute the used cartridge, meaning we can wash it out and we can find out what's in it. So we can look directly at what we've gotten out of it. And we'll be doing that in real time.
Absolutely. In terms of overall, and just lastly, anything else important to point out about the device, doctor, that you haven't previously stated?
Well, yeah, I mean, we're talking about viral disease today, but number one, as I said, we have data on multiple different viruses, either in the lab or in humans or both. And so not only do we expect to be able, or do we hope to be able to intervene in this pandemic, we hope to be a part of the response to future pandemics. And unfortunately, we're going to be seeing this over and over again for a whole bunch of reasons that everybody has talked about: deforestation, overpopulation, all people getting closer to bats and other kinds of vectors for these types of viruses. And the Hemopurifier is a very broad-based therapy. That is, any viral infection theoretically can be treated with it. So we're focused on that. But the other point here is I mentioned that exosomes are involved in cancer, and we're very actively involved in that. We have an open investigational device exemption for cancer, for the treatment of patients with head and neck cancer. And we're starting up a trial at the University of Pittsburgh Hillman Cancer Center, which is one of the top cancer centers in the country. And what we're looking at is we're looking at combining the Hemopurifier treatment with a drug called pembrolizumab or Keytruda that you've undoubtedly heard of because it's the largest selling drug in the world right now. And what Keytruda does is it takes the brakes off the immune system. So our immune system can recognize malignant cells because they have mutations in them that make them different from normal cells. So our immune system can recognize cancer cells. And in you and me sitting here, if we're healthy and we don't have cancer, those kinds of cells are being generated all the time, but just small numbers of them, the immune system keeps them under control. For a number of reasons, when patients get cancer, particularly advanced cancer, they become profoundly immunosuppressed. Their immune systems do not work against the tumor. And what pembrolizumab or Keytruda does is it takes the brakes off that response so that their immune system can actually kill the cancer cell. And these drugs are extraordinary. They're taking some patients with widespread metastatic disease, melanoma, lung cancer, bladder cancer, other cancers, and putting them into remission for 5 years or even longer. And these are patients who would have died in 6 months prior to the advent of these drugs. But unfortunately, they only work in a minority of patients. About 20 to 30% of the patients respond, and the rest of them don't. So the improvements you see in survival are driven by a minority of patients. It's now been shown that one of the primary mechanisms by which patients do not respond to those is mediated by exosomes. So what we're going to do is we're going to take the Hemopurifier and treat patients with head and neck cancer who are going to be started on Keytruda before they get to Keytruda to see if we can lower the number of circulating exosomes and improve both the percentage of patients who respond and the duration. If we're able to show that in head and neck cancer, it should generalize to multiple different solid tumors where Keytruda is active.
Absolutely. Well, we want to wish you the best of luck with the trial, and please keep us posted on everything. And thank you very much for talking about this important topic today.
Thanks so much. It was a pleasure to be here.